We think biology is one of the neatest things on the planet. That's why we're making this video. We want everybody to know how neat biology is. We're going to talk about an experiment that involves the replacement of a chromosome in a eukaryotic cell, specifically Saccharomyces cerevisiae, which is the yeast used in brewing beer. Brewer's yeast has 16 chromosomes, and chromosome 3 is the one that is being experimentally created. This is one of the shortest chromosomes of the yeast, and it determines yeast's mating type. The purpose of this experiment was to create a strain of yeast with a man-made chromosome 3, which would have specific properties that could be manipulated to exhibit certain traits. Chromosome replacement has been done before in prokaryotes, like E. coli, but nobody had ever made a eukaryotic chromosome and placed it into a cell.
This chromosome synthesis, conducted by a team of international scientists, began with a computer program to design the nucleotide sequence before ever starting with real nucleotides. Once they had the chromosome sequence planned, they built its pieces using a process called the polymerase chain reaction method. The polymerase chain reaction method, you start with your DNA strand. You pick a region that you wish to have in order for your building block. Once you have this region, you denature the DNA by putting it in high temperatures. This breaks the hydrogen bonds that are between each strand, or the nucleotides on each strand. Once you do this, you add something called anneal primers. These connect to the sites on the boundaries of where you wish to have your DNA come out. When you put this in the presence of polymerase, they move in opposite directions of each other. And when you do this, they actually create complementary base pairs from the point at which the polymerase anneal primer group started and ends at the end of the molecule. This happens for both ends. So, they then denature each of these again. This one is this one, and this one is this one. And they add anneal primers as well as polymerase. But this time, they go in opposite directions. So, when you're done, you end up with two DNA molecules that the exact length that you wish and are also have the nucleotides that you wish to have.
The polymerase chain reaction method yields two copies of the desired segment of DNA. With these segments, they made the building blocks to create the Sen3's chromosome. They placed groups of building blocks into the chromosome 3 in 11 steps. In this figure, which shows the first two of these 11 steps, you can see that 4 and 9 building blocks were placed in steps 1 and 2 respectively. After 11 steps, the sections of designer DNA replaced the corresponding sections of the chromosome, and all of chromosome 3 was replaced. They called this new chromosome Sen3. To confirm that the new yeast with Sen3 chromosome were as viable as normal yeast, the two yeasts were tested in several different environmental conditions. As you can see, the wild-type brewer's yeast reacts very similarly to Sen3 under different temperatures, as well as under conditions of low and high pH. Further, the different cells reacted very similarly to the same growing conditions. This holds true in both colony number and loss rate. In essence, the difference in fitness of Sen3 and the wild-type yeast was negligible.
The total synthesis of the Sen3 chromosome represents a major step toward the design and complete synthesis of a novel eukaryotic genome structure. Jeff Buka from New York University Langone Medical Center, who worked on this study, is quoted as saying that yeast cells carrying synthetic chromosomes are remarkably normal. They behave almost identically to wild yeast cells, only they now possess new capabilities and can do things that wild yeast cannot. This is a step towards being able to synthesize other living cells for use in the biotechnology industry, which is extensively using yeast as a platform for producing all kinds of useful medicines and vaccines, or to produce an antibiotic that can turn agricultural waste into biofuel like alcohol and biodiesel. Rapid advances in synthetic biology suggest that it will soon become feasible to engineer new plant and animal genomes with synthetic chromosomes encoding desired functions and phenotypic properties. I know what you're thinking now. This is some weird science.